thesis is metagenomic and genomic analysis of modern freshwater microbialites unmasking a community of complex metabolic potential. So this kind of started off with a conversation with Curtis when I joined the group about 4 years ago, and he went into the office and was like, well, Rick, you know a lot about sequencing, you know a lot about reconstructing various things in genomes. Have you ever heard of a stromatolite? And the answer was absolutely not. I had no idea what that was, or what a microbialite was. And so for the last 4 years, I've tried to dive deeper into this very complex ecosystem. And today I'm going to try to show you and talk about my favorite ecosystem. So we'll start off with, what is a microbialite? So a microbialite is defined by Ewan Moore as an organic sedimentary deposit that's excreted as a result of a benthic microbial community. And you're like, what is that? Very kind of convoluted. So basically you have a complicated microbial community interacting with abiotic processes, forming a solid structure as a result of this. So that's really what they are. So it's a biotic and an abiotic community that are functioning together to form some sort of hard structure. And so they're classified geologically. So the first type of microbialite is a stromatolite. So strombus meaning layered. So you have a layered rock deposit. And normally these comes in two major isoforms. They come as either calcite or hydromagnesite, which is a magnesium carbonate, and calcite being a calcium carbonate, so chalk, Mount Rushmore. These are all examples of calcium carbonate. You have the dendrolite, so basically a dendritic-like form structure that forms inside the microbialite. This is another classic example. The one that I see the most of in freshwater are these things called thrombolites. So thrombus meaning clot. So basically you have these clotted structures that are being squished together, forming these kind of hard structures. From there, you have this thing called an oncolite, which is basically a type of stromatolite, but so you have the layered and the lamination, but instead of forming in kind of a flat layer, it forms in a circle. So these are the four major forms of microbialites we see today in modern microbialites. So what's kind of cool is that, like I said, they're made of calcium carbonate. So if you take 100 grams of microbialite, 12 grams of pure atmospheric carbon was trapped in that microbialite. So it has a lot of interesting aspects as far as not only the idea of origin of life, and I'll talk about that in a minute, but even modern day. These things are basically banks of CO2 gas. So I'm going to walk you through geological time, and this is of course not my data. This is a friendly gift from Dr. Sean Crow, and he just published a Nature paper, and I had to put back all the timelines of the origin of microbialites based on his paper. And so what I have here is basically time of the Earth in billions of years on the x-axis, and then on the y-axis I have percent of atmospheric levels of oxygen, so this is percent of levels that we see today. Okay? And he basically went through with chromium isotopes and basically measured the amount of oxygen throughout geological time. So let's start from the beginning. So in the beginning, we had Earth kind of forming around 4.5 billion years ago, and this is called the Hadrian period. Hadrian meaning hot or hell, basically. And lots of collisions, lots of stuff going on. A moon formed in this time frame, so there's of course no oxygen present in the atmosphere. Go a billion years later, the Earth starts to, less bombardment occurs, the Earth's planet starts to cool, and we're getting water forming on the surface of the planet. Once again, no oxygen in the atmosphere. Around this time, something magical happened, and is that you have the beginning of these small organic molecules such as amino acids and RNA that began to form. From this kind of complexation, they formed DNA, which then, with protein and membranes, formed the first cells. From these were the first biochemists, and all of these biochemistries, such as methane, methanogenesis, sulfate reduction, were anoxic, in the presence of without oxygen. So the first kind of beginning of life on the planet and the first kind of biological systems that formed geochemical cycles were all anoxic. From there, we have the first microbialites at around 3.5 billion years ago. And there's a little bit of a spike in present atmospheric levels at this time, but this is within standard error, so we believe that there's still no oxygen in the environment at this time. And so the oldest rocks that gives us the litmus test, the signpost that said there was life on the planet, were these structures. Back 3.5 billion years ago, so were found in the Warnaid group deposits in southern Australia. Well, everyone talks about little green men from Mars. I want to talk to you about the little green men on Earth, right? So cyanobacteria. These were the little green men that gave us the planet we have today. They basically terraformed our planet by the birth of oxygenic photosynthesis. With this oxygenic photosynthesis, you have this huge spike in present atmospheric levels, and you have the birth of modern microbialites. So the ones I talk about today are in the presence of cyanobacteria, because once cyanobacteria happened, basically 1,000 new minerals formed. You had an oxidation of the plant. All rocks prior to that we term the ancient stromatolites, so anoxic stromatolites. Over time, you had eventually a drop in atmospheric carbon, and once again it started to build up again through the presence of cyanobacteria. Actually, this is kind of the normal time. We want to think that cyanobacteria and oxygen really happened on the planet. But because of Sean's work, we're able to put that timeline back around another 300 million years. So over time, more oxygen built in the environment, leading to this thing called the Great Oxidation Event at 2.4 billion years ago. This is the greatest mass extinction event in the history of man, in the history of the Earth. 99% of 99.9 with 100 nines of all species on the planet disappeared in this period. Of course, you know, if you're thinking about the stromatolites as a time frame, they've been around for a long, long time. At around, you know, basically this time you had the first alga, you had, around the beginning of the Precambrian expansion, you have a general decline of stromatolites, and decline of microbialites as a general whole. And we think that basically through eukaryotic grazing, that's what led to kind of the oceanic decline of stromatolites all over the planet. And that's been kind of the central dogma, even to this day. And to put us in perspective, we're like right here, just at the very end of geological time. But if you to get anything from this, I want you to leave with this, is that 85% of Earth's history, microbials have been present, and they represent the oldest continuous biological media process on the planet. There is nothing older than this process. So people were interested in this because of the whole origin of life, the sequestration of carbon, atmospheric carbon specifically. And so back in '73, a guy wanted to ask us, like, how common is this phenomenon? And actually he found this phenomenon of basically taking microbes and putting them in the correct media, in the correct abiotic conditions, actually be very common. Up to 200 isolates that you can find right out here in the soil that could actually make calcium carbonate. So this makes a larger problem. So you have this very complex community, and it seems like everybody can do it. So it makes it very complicated to try to study this as a whole. So from there, and this is a beautiful thrombolite from Kelly Lake, actually, and this thrombolite is as big as this table right here. Okay, this is huge, okay? And so this is kind of the current model from Dupois about how we think that microbialites are kind of forming in these kind of complicated ecosystems. So as I mentioned, you have to have abiotic factors that are very, very important. You're not just going to have the right microbial community and put it at 5 pH and expect the rocks to form. They're just not going to do that. So you need an alkaline water column, so basically 8 pH or greater. You need dissolved cations, so either magnesium or calcium, and then dissolved carbonate. As you look at these as a structure, you have these EPS layers, so these are extrapolysaccharides that are made by a filamentous cyanobacterial mat. And as you go through the mat, you have layers of aerobic all the way down to anaerobic, so various niches where various complicated microbial communities can live and exist. In the case of Pavilion Lake and the ones that I study, it's basically aerobic all the way through the water column. However, if you look at something like the Highborn Cay in the marine system, you have stratification of various nitrate and sulfate, these kind of things as well. So, once again, little green men, little green bacteria, basically. Photosynthesis is the keynote part of this, because not only, especially oxygenic photosynthesis, because they basically split water and from light can basically they can trap through photosynthesis and then through the dark reaction trap carbon. This process within the correct abiotic chemistry forms calcium carbonate. And so who are the main green photosynthesizers in these ecosystems, and these are cyanobacteria and eukaryotic protists. All right, there's another kind of interesting part that is more of Sue's Baldwin's kind of thing, and she talks about these guys a lot in metals and these other systems, but I really enjoy them in looking at them in microbialite ecosystems. And these are the assimilatory sulfate reducers. So these are commonly found in the delta proteobacteria. They're also found in Nitrospira. And what they're doing is they're basically using sulfate through anoxic metabolism, and in the process of doing that, they're basically trapping calcium carbonate. And so when you have those anoxic layers inside the mat, this is where a lot of the infilling of the carbonate precipitation is occurring. This is the oldest one, so the ancient microbialites. That's how we know that there was biological activities through looking at sulfur isotopes in these ancient stromatolites. So these have been important since 3.5 billion years, and they're still important today. All right, so map of Earth. And so initially when I first studied this, was given this as a question, is like, well, there's only a couple places. There's one over here in the Bahamas, and there's one over here in Shark Bay, Australia. And the oldest fossils, like I mentioned, are found in Shark Bay, Australia. But as I started looking around, I found them everywhere. And these are ones that have active microbialite forming communities all over the globe. And I guarantee that if you look throughout in areas of Africa and parts of Eurasia, you would find more of these things. Actually, Pavilion Lake, the ones I'm going to talk about today, they've been known since 1996, and they were called freshwater corals. So there was divers that went in and said, hey, you know, what's with all the corals? Well, they're not corals. They're microbialites. And so everything from freshwater, marine, hot springs, hypersaline, as long as there's an alkaline pH system and dissolved cation and carbonate, you get these structures, even at 70 degrees. Actually, the one over here in New Zealand is 80 degrees. So as far as temperature goes, not a problem. So like I said, mentioned today, I'm going to talk about Clinton Creek. And Clinton Creek is a really interesting one because it formed very rapidly. And it's basically from a mine tailing site. So basically it was an asbestos mine. They dug it out and water filled in. And then over 25 years, you have these basically no bigger than about that big tall structure. So the fastest growing calcite forming microbialites on the planet. But kind of anthropomorphic in the fact that we kind of made this site through kind of dredging. And then the one that I talk about most about is Pavilion Lake. So Pavilion Lake, you know, get a bigger thing about Pavilion Lake in a second. So methods. So basically from there, we basically grab around 100 liters of water, 10 grams of microbialite, and then 10 grams of sediment. I extract DNA, make my own libraries, go 4x4 on Illumina, and then sequence. So spend most of my time in the dry lab in the computer. So after it comes off the sequencer, I have some raw data, and it's pretty rough. So I have to kind of trim it and clean it up and take out like phi-X reads and these kind of things. So then I have to clean the data, and then I assemble it with Ray. And Ray is a metagenomic assembler. And what it does is it's going to take all these various platforms and kind of take if it doesn't find a match, it's just going to leave it alone and not assemble it. And it won't form chimera, something like Nuclear or one of these other assemblers can do as well. From there, I'll do annotation with either MGRAS or metapathways. Of course, metapathways doesn't have a cool icon that I can put up here. And then further analysis with R and then stamp. Okay, so with every great scientific thing, you have to start with a question. And so my first question, and Curtis and our first question was, does the metabolic potential and the community structure differ between the microbialite and the nearby sediment in the water? And the reason for that is that, well, you have these things that are surrounded in water. Basically, Pavilion Lake is surrounded completely in water. And the question is that a lot of geologists would say, well, you know, basically there should be no difference between any of them, right? And there should be no change because basically it's only the abiotic chemistry. Nothing should change. There should be no selection or anything that's going on in that. But we wanted to kind of investigate that a lot further. So first kind of question was in Pavilion Lake was that there's these unique morphologies as a function of depth. So you have basically lettuce beds, cabbage heads, you have conal structures, and then these large artichoke structures. And the first question was, well, is there a specific microbial community structure as a function of depth? And right here I'm going to show you that there isn't. And basically what we have here is a simple rank abundance curve, but basically from 0 to top 35 bacterial taxes looking at with RefSeq, so we're talking about the protein coding genes, using BLAT, which is BLAST-like search algorithm, so we're basically using a database to tell us what is going on in the metagenome, and then we have relative proportion on the y-axis. And we can see that basically as you go down in depth, regardless of the morphology of the microbialite, that basically alpha proteobacteria And the first 4 basically are the proteobacteria. Around 90% of the community in Pavilion Lake is of proteobacteria. And of course, our little green bacteria, cyanobacteria, around 4th or 5th. So no unique microbial community structure as a function of the unique morphology at update. So kind of a bummer. But we did answer our original question, which was, and of course this is looking at the functional relevance. So basically we're taking the functional genes and from the seed classification, and we're using BLAT. We're displaying this in a PCA from here with the base Curtis distance of 1,000 replications. And we can see right off the bat, and of course in green, we have microbialites. In red, or kind of off red, we have sediments. And then in blue, we have the water column. So you would expect that at 10 meters, if there's basically a lot of mixing going on and the communities don't have unique microbial community structure, you would expect that the sediments from 10 meters and the water from 10 meters and the microbialites would all be the same. That's not the case. Is that basically the microbialites are more similar to each other than they are to the surrounding water and the surrounding environment. So that's kind of interesting, because that basically says that, hey, you know, there's something unique about the microbialites that's different from the surrounding environment. Okay. So we went further with that, and we basically did a seed ANOVA, and we took that another step and did an ANOVA on this, and we basically found the same thing with the functional pathways, which are basically the microbialites are distinct from the surrounding environment. So that was basically answered our first question without a doubt, that microbialites are distinct from their surrounding environment. And that is unique because if they were all the same, it wouldn't really mean as much as if they're different from each other. So there's a unique community that's specific to microbialites that's different from the surrounding environment. Okay. So then the next question we had, which was which carbonate precipitating pathways are overrepresented in microbialite versus nearby sediments. And so we're going back to Clinton Creek. So Clinton Creek does this really, really fast. Okay? Does it faster than any other microbialite site on the planet. And in the case of Pavilion Lake, it takes thousands of years for these structures to form. So if you're trying to have a real-world example of how these things form and function, it'd be great to have something that could do it in your lifetime. So that's the reason for looking at this. So it's not as it's very different. Like I said, it's it's a different type of site. You don't have the kind of water covering for the most part, but you do have sediment surrounding. So we wanted to ask the same question again: is the sediment and the microbialite the same as far as the microbial community structure? So once again, we just have some basic microbial taxa. We have the rRNAs, so all the ribosomal RNAs, and then the open reading frames. So these are the protein coding genes for both the microbialite and the sediment. And basically, right off the bat, you can see it, is that the cyanobacteria are different. And cyanobacteria are essential for the formation of the structures. You cannot have, without the cyanobacterial mats, you do not have the structures. And right off the bat, you have no ribosomal RNAs, and the taxa that we do have for protein coding genes for the sediments are non-filamentous. They're only platonic or single cellular. All the filamentous genera are in the microbialite. So once again, and then from talking about it a little ago, what are the kind of major carbonate precipitating pathways that are involved in the formation? So cyanobacteria, that's a good one to start with. So this is a Fisher's exact test in STAMP. And what I wanted to know is basically take the sediment versus a microbialite using the seed subsystem functional gene classifications, which metabolic pathway at the top level, the highest level, relating to carbonate precipitation is most highly represented. And right off the bat, it's significant enough, but photosynthesis is more significantly represented in the microbialite over the sediment. So more cyanobacteria, more filamentous cyanobacteria, more photosynthesis, likely relating to also our isotopic data that suggests there's an isotopic enrichment of the carbonate in the microbialite. So they all work together. And now I'm kind of switching gears a little bit, talking a little bit less about the systems and more about this other kind of unique thing is basically we do our heterotrophic isolates represent new species and which pathways are involved for the colony pigmentation. So the idea was is that we go out and we took a 20 meter microbialite from Pavilion Lake, We wanted to culture some of these guys so we can kind of figure out and kind of tease out which some of these members that could actually precipitate carbonate. And we really couldn't find any of those that really precipitated carbonate well. We did find these two unique isolates. And these two unique isolates, of course, is our yellow isolate of Argococcus, which is high GC. It's a rare member. There's only been about 7 that have been cultured. And then we have our orange isolate, Exogobacterium, which is, actually very interesting. There's about 12 genomes that have been just recently published, actually in the process of doing the thesis, and has a lot of industrial aspects to it. It's an extreme affair. So, once again, we have basically a dot plot to compare the 2 known genomes. So basically our Argococcus species. There's only 2 representative genomes, and I tried 16S. I tried multiple locus sequence typing to try to say, okay, are they different? And the question was, does Argococcus, strain RW1, is it different from any other strain that's currently known? And for the most part, it is, because you would expect on a genome plot like this, if they were identical, that this would be a straight line, as well as there's many gaps. So what you're looking at here is that you basically have the genome of Argococcus lahalensis on the x-axis, and then Argococcus strain RW1 on the y-axis, and you have here as well, you have a MAVE plot for homology, and basically there's many gaps. We then took that to the functional potential and asked the question, okay, from the amino acid level, are the 2 of them different? And basically they are. We would assume that if they were the exact same species, they'd be 95% identical. Okay? However, they're not. And what's also really interesting is that greater than 55% of the open reading frames are hypothetical or unknown. Right? If you compare that to E. coli, it's less than 30% of E. coli genome is not known. So these guys are new and novel in that respect. So then the last one, it's, like I said, very important industrially. And we wanted to see where our isolate landed, and basically because it's less than 97% like any of the other known species, we were able to show that with just 16S that our isolate strain was different from the rest. Of course, we did metabolic potential analysis and these kind of things, but our maximum likelihood with excluding the bootstrap basically tells us that our strain is a new species. All right. Won't get into a lot of time to this today. Basically, the next question was, well, how are the yellow and how are the orange? So our orange guy is basically through the C30 carotenoid pathways. It has all the genes predicted in the genome for this pathway to make and we predict it to be a dineuronorspine acid that basically makes the colony pigmentation. And then for our yellow strain, it also has all the predicted genes for making xanthan. And it could be also making another yellow pigment, but further experiment evidence is needed to validate these parts of the genome. So what is the take-home message? So communities of microbialite appear distinct from the neighboring environments. This is new. No one else has ever done this or even asked that question. The next thing is that photosynthetic induced alkalization, so the driving of our little green bacteria basically are the mechanism that's providing rapid formation in Clinton Creek. And then carotenoid pathways are likely responsible for colony pigmentation in our isolates. So, quick announcement. I wanted to definitely thank my professor, Dr. Curtis Suttle, for 4 years of enrichment and hunting around various parts of BC, and the Suttle Lab, and my committee, Sue Baldwin, who's here with us today, Tom Beatty and Stephen Hallam, various folks in the Beatty Lab, Dipple Lab, and Anna Harrison and Ian Power, who helped me with the Clinton Creek stuff, various brilliant bioinformaticians such as Nils and Stillion, and folks in the Foster Lab, and the various people that have kept me sane through the last 4 years of trying to figure this very complicated thing out. So I thank you very much for your time.